Welcome back, class. This is Silvana O'Coney with Lecture 8, Antimicrobial Chemotherapy. Now, we know that the control of microorganisms is critical for preventing disease and treating disease. But antimicrobial compounds are also incredibly important tools used in research and also in industry. Now, we've looked at the use of chemical and physical agents, and when disinfecting or sterilising an inanimate object, one naturally has to use procedures that don't damage the object itself. The same is true for treating living organisms. The most successful antimicrobial compounds will interfere with vital processes that differ between the microorganism and the host. Thereby, it seriously damages the target microorganism but harms the host as little as possible. So this lecture is going to introduce the principles of antimicrobial chemotherapy, and briefly we'll look at the characteristics of selected antibacterial, antifungal and antiviral compounds. Chemotherapeutic agents. These are chemical agents that are used to treat disease. Ideally, these agents should destroy pathogenic microorganisms or inhibit their growth at concentrations low enough to avoid undesirable damage to the host. Most of these chemotherapeutic agents are antibiotics. These are microbial products or their derivatives that can kill susceptible microorganisms or inhibit their growth. Drugs such as the sulfonamides are sometimes referred to also as antibiotics, although they're actually synthetic chemotherapeutic agents. They're not microbially synthesized. The modern era of chemotherapy began with the work of the German physician Paul Ehrlich. Ehrlich was fascinated with dyes that specifically bind to and stain microbial cells. He reasoned that one of the dyes could be a chemical that would selectively destroy pathogens without harming human cells. A magic bullet. By 1904, Ehrlich had found that the dye called trypan red was active against a microorganism that causes African sleeping sickness and could be used therapeutically. Subsequently, Ehrlich and his young Japanese assistant, pictured here, tested a variety of arsenic compounds on rabbits that were infected with syphilis. And they found a compound that was active against the syphilis microorganism. This compound was made available in 1910 under the trade name Salvarsan, and it paved the way to the testing of hundreds of compounds for their selective toxicity and therapeutic potential. Then in 1927, the large German chemical company began a long-term search for chemotherapeutic agents under the direction of Demark. He screened a vast number of chemicals for other magic bullets, and he discovered a new dye that completely protected mice against pathogenic bacteria such as streptococci and staphylococci without any apparent toxicity. This paved the way for the discovery of the sulfonamides or sulfur drugs. During the 1920s, the Scottish doctor Alexander Fleming found that human tears contain a naturally occurring antibacterial substance, and he called this substance lysozyme. This substance unfortunately had very little therapeutic activity because it couldn't be isolated in large enough quantities and wasn't effective against many microorganisms. However, it did prepare Fleming for the discovery of penicillin, which was the first true antibiotic to be used therapeutically. Penicillin was actually discovered in 1896 by a 21-year-old French medical student. But his work was forgotten until Fleming's accidental rediscovery of the antibiotic in September 1928. After returning from a weekend vacation, Fleming noticed that a petri dish of Staphylococcus also had a mould growing on it, just like the one pictured on this slide. And just like the lysozyme he had discovered years before, there were no Staphylococcus colonies surrounding the mould. Although the precise events are still unclear, it's been suggested that a Penicillium notatum fungal spore had made its way onto the petri dish before it had been inoculated with the bacteria. The mould apparently grew before the bacteria did, and it produced a substance which was later named penicillin. The bacteria nearest the fungus, so nearest that colony of mould, were killed. Fleming correctly deduced that the mould contaminant produced a diffusible substance, which he called penicillin. And in subsequent studies, he showed that this substance could diffuse through agar, so that even small amounts of it extracted from broth cultures could kill several pathogenic bacteria. However, Fleming could not demonstrate that penicillin remained active for long enough to destroy pathogens, and he didn't think it could be developed any further. He dropped the research. So it wasn't until 1939 that Howard Florey, a professor of pathology at Oxford University, he was in the middle of testing the antimicrobial activity of many different substances when he came across Fleming's paper on penicillin. So he set about with one of his co-workers, Ernst Chain, set about culturing the mould and purifying the penicillin. And they were aided in this by the biochemist called Norman Heatley. When the purified penicillin was injected into mice that were infected with streptococci or staphylococci, nearly all the mice survived. So this success was reported in 1940, and subsequent human trials were equally successful. Fleming, Florey and Chain received the Nobel Prize in 1945 for the discovery and production of penicillin. This discovery of penicillin stimulated the search for other antibiotics. Selman Waxman announced in 1944 that they had found a new antibiotic called streptomycin. And this antibiotic was produced by the bacterium Streptomyces griseus. The discovery arose from very careful screening of about 10,000 strains of soil bacteria and fungi. The importance of streptomycin cannot be understated, as it was the first drug that could successfully treat tuberculosis. Waxman received the Nobel Prize in 1952, and this success led to a worldwide search for other antibiotic-producing soil microbes. Microorganisms producing chloramphenicol, neomycin, terramycin and tetracycline were isolated then by 1953. So, as Paul Ehrlich so clearly saw, to be successful, a chemotherapeutic agent must have selective toxicity. That is, it must kill or inhibit the microbial pathogen while damaging the host as little as possible. The degree of selective toxicity may be expressed in terms of 1, the therapeutic dose, which is the drug level required for treatment of a particular infection, and 2, the toxic dose. That is, the drug level at which the agent becomes too toxic for the host. That gives us what's called the therapeutic index. That is the ratio of the toxic dose to the therapeutic dose. The larger the therapeutic index, the better the chemotherapeutic agent is. So a drug that disrupts a microbial function that's not found in eukaryotic animal cells often has a greater selective toxicity and a higher therapeutic index. For example, penicillin inhibits bacterial cell wall synthesis, but has very little effect on host cells because they lack cell walls. Therefore, the therapeutic index of penicillin is high. A drug may have a very low therapeutic index because it inhibits the same process in the host cells or damages the host in other ways. And the undesirable effects on the host or side effects are of many different kinds and may involve almost any organ system. As you can imagine, drugs vary considerably in their range of effectiveness. Many of them are narrow spectrum drugs, that is, they're only effective against a limited variety of pathogens. Others are broad spectrum drugs that attack many different kinds of pathogens. Drugs may also be classified based on the general microbial group they act against: antibacterial, antifungal, antiprotozoan, antiviral, and so on. Some agents can be used against more than one group. So, for example, the sulfonamides are active against both bacteria and some protozoa. Chemotherapeutic agents like disinfectants can be either cidal or static, as we've seen previously. Remember, static agents reversibly inhibit growth. If the agent's removed, the microorganisms will recover and grow again. Although bacteria and fungi are able to naturally produce many of the commonly used antibiotics, many of the important chemotherapeutic agents are actually synthetic. That is, they're manufactured by chemical procedures independent of microbial activity. An increasing number of antibiotics are semi-synthetic. That is, they're natural antibiotics that have been structurally modified by the addition of chemical groups. And this is done to make them less susceptible to inactivation by the pathogens. Some examples include ampicillin, carbonicillin, and methicillin. In addition, many of the semi-synthetics have a broader spectrum of activity than does the parent molecule. This is particularly true of the semi-synthetic penicillin, for example, ampicillin, versus the naturally produced penicillin G and penicillin V. This table summarises the microbial sources of some of our antibiotics. First one on the list is the bacterial genus Streptomyces. Streptomyces are found predominantly in soil, and they produce 2/3 of all known antibiotics. One example of an antibiotic they produce is amphotericin B. This is an antifungal that is fine when it's ingested orally, but it's extremely toxic when given intravenously. So it has a very low therapeutic index. Some idea of the effectiveness of a chemotherapeutic agent can be obtained from the minimal inhibitory concentration, or MIC. This is the lowest concentration of a drug that prevents growth of a particular pathogen. On the other hand, the minimal lethal concentration, or MLC, is the lowest drug concentration that kills the pathogen. A cidal drug generally kills pathogens at levels only 2 to 4 times the MIC, whereas a static agent kills at much higher concentrations, if at all. There are 2 techniques that are routinely used to determine MIC and MLC. The first of these tests is the dilution susceptibility test, and this test can be used to determine both MIC and MLC values. Antibiotic dilution tests can be done in both agar and broth. In the broth test, a series of broth tubes containing different concentrations of an antibiotic is prepared and inoculated with a standard density of the test organism. The lowest concentration of the antibiotic resulting in no growth after 16 to 20 hours of incubation is the MIC. The MLC can be ascertained if the tubes showing no growth are subcultured into fresh medium lacking the antibiotic. The lowest antibiotic concentration from which the microorganisms do not grow when transferred to fresh medium is the MLC. The second technique is the disc diffusion test. The principle behind this assay technique is quite simple. When a disc impregnated with an antibiotic is placed on agar that's been previously inoculated with the test bacterium, the antibiotic will diffuse radially outward through the agar, producing an antibiotic concentration gradient. So the antibiotic will be present at high concentrations near the disc and affects even minimally susceptible microorganisms. Keep in mind that resistant microorganisms will grow right up to the disc as they're not affected by the antibiotic. As the distance from the disc increases, the antibiotic concentration decreases and only more susceptible microorganisms are harmed. A clear zone or ring is present around an antibiotic disc after incubation if the agent inhibits bacterial growth. The wider the zone surrounding a disc, the more susceptible the pathogen is. Zone width is also a function of the antibiotic's initial concentration, its solubility, and its diffusion rate through the agar. Currently, the disc diffusion test most often used is the Kirby-Bauer method, which was developed in the 1960s and also is the method that we'll be using in crack class. It involves using an inoculating loop or a needle, which is used to pick up 4 or 5 isolated colonies of the bacterial pathogen, which is growing on agar, and this is then used to inoculate a tube of culture broth. The culture is incubated for a few hours and then it's diluted. A sterile swab is dipped into the bacterial test suspension and used to evenly inoculate the entire surface of an agar plate. After it's dried for about 5 minutes, the appropriate antibiotic test discs are placed on it with sterilised forceps. The plate is incubated, and then after 16 to 18 hours of incubation, the diameters of the zones of inhibition are measured to the nearest millimetre. So sensitivity to the antibiotic or resistance to the antibiotic are determined using tables that relate zone diameter to the degree of microbial resistance. Here's an example of a table that shows the inhibition zone diameters of selected chemotherapeutic agents. Let's look at one example. In the column on the left-hand side, go down until you find penicillin G with staphylococci. Then we'll look across at the zone diameters. So in the column with the heading resistant, you can see that if the inhibition zone diameter is less than or equal to 20 millimetres, then the microorganism is said to be resistant to that particular chemotherapeutic agent. Move across now to the column on the right-hand side. And this tells us that if the zone of inhibition has a diameter that is greater than or equal to 29 millimetres, then the microorganism is susceptible to penicillin G. The e-test is very similar, but it's particularly convenient for use with anaerobic pathogens. Each strip that you can see here on this agar plate contains a gradient of a particular antibiotic, and it's labelled with a scale of minimal inhibitory concentration values. The lowest concentration in the strip lies at the centre of the plate. After 24 to 48 hours of incubation, an elliptical zone of inhibition appears. And you can see that elliptical zone around the outer edge of each e-test strip on this plate. As shown also in the figure and labelled with the red arrow, the MIC can be determined from the point of intersection between the inhibition zone and the strip's scale of MIC values.